When the action potential reaches the end of the cell at the synaptic bulb or the axon terminal, it is going to open up another type of channel called a voltage-gated calcium channel. Here it is right here. We know that calcium is higher on the outside of the cell, so the calcium will move in. When it does, it triggers the mechanism of exocytosis. In the neuron, what happens is the synaptic vesicles filled with neurotransmitter are going to fuse with the membrane and release the contents into the synapse. What happens then is that these transmitters are going to diffuse across the synapse and bind to receptors on the next cell. So this would have been the presynaptic and this is the postsynaptic. So we are now back to the beginning of our story where the neurotransmitter binds to the postsynaptic receptor. It is then going to open up a chemically gated channel and, for example, sodium may come in, and in this case we would get an IPSP, or perhaps it's opening up a potassium channel, in which case potassium is going to leave and we would get an IPSP. Once again, which channel opens up or closes depends on the interaction of that neurotransmitter with the receptor. Next diagram, we'll take a closer look at what's happening at the postsynaptic cell. Now we're essentially back at the beginning of the story, where we have the release of transmitter from the presynaptic cell stimulating a postsynaptic cell. So let's take a look here in a little bit more detail. What I'm trying to illustrate is that we have three different cells. Here is one that is releasing neurotransmitter A. Okay, here another cell is releasing a different type of neurotransmitter, and here we have a third cell releasing a different type of neurotransmitter. Each one of these neurotransmitters is binding to a specific receptor. And remember that one neurotransmitter can actually bind to different subtypes of receptors, so it gets quite confusing. Depending on the receptor, a particular chemically gated channel will be open, or potentially closed, but let's just try to keep it simple and think of it as opening the channels. So here we have a chemically gated sodium channel, which we discussed before is going to cause a depolarization and an excitatory postsynaptic potential. Alternately, here we have a chemically gated potassium channel, which is going to cause a hyperpolarization and an inhibitory postsynaptic potential. Another ion that can move to cause an inhibitory postsynaptic potential is chloride. If chloride moves in, it's bringing negative charge into the cell, making it more negative, and again causing a hyperpolarization and an IPSP. Okay, remember from before that the overall result of integration is that we're going to add all of these up, and of course it depends on the net result as it reaches the axon hillock, remembering decremental conduction, so the overall response is going to be summated, and this response, this graded response, will either bring the cell to threshold, in which case we're going to generate an action potential, which will then propagate down the cell, or it will not, in which case nothing happens. Okay, the next slide, I'm just going to finish off talking about how we get rid of this neurotransmitter in the synaptic cleft. There's three basic mechanisms by which the action of these neurotransmitters is terminated. Remembering that as long as these neurotransmitters are in the synapse, binding to the receptors and opening up these channels, these chemically gated channels, okay, the ions are going to continue to move and continue to elicit postsynaptic responses. So if we want to terminate the stimulation, which is just as important as initiating it, we have to get rid of the neurotransmitter. So the three ways are, here we have what represents is an enzyme, okay, the E. We're going to enzymatically break down the neurotransmitter, and it will now no longer be able to stimulate the cell. Another way is that we could have reuptake. So the neurotransmitter gets taken back up into the presynaptic cell and will be recycled to use again. Third mechanism is simple diffusion out of the synapse and again terminating its action.